Hello, and welcome to Informed in 30, a new series from the National Brain Tumor Society designed to give you, our patients and caregivers, everything you need to know about cutting-edge advancements in brain tumor research and treatment. Our topic today is innovations in precision medicine. My name is Rachael Kittleson, and I'm the Director of Community Relations here at the National Brain Tumor Society. I also represent the brain tumor community as a former caregiver to my daughter who passed away from glioblastoma in 2017. Joining us today is Dr. Kirk Tanner, National Brain Tumor Society's Chief Scientific Officer, and Dr. Patrick Wen, Director of the Center for Neuro-Oncology at Dana-Farber Cancer Institute and Professor of Neurology at Harvard Medical School. Thank you both for joining us today.
Yeah, thanks so much, Rachael. I'm very happy to be a part of this series. I think it's really important, and we're really excited to be able to bring this such important information to our brain tumor community.
Thanks so much for having me. It's a real pleasure to be here.
No, thank you, Dr. Wen. We really appreciate it. So what I think would be good is that let's start out with an overview of precision medicine and, what does that really mean to patients? How does understanding the genetic and molecular characteristics of brain tumor guide treatment strategy? Dr. Wen, as a real dedicated to patients and treatment, and as a visionary leader in the field, we're really so fortunate to have you today to be able to introduce this topic to our audience. So thank you very much, Dr. Wen. I'll turn it over to you.
Thanks so much, Kirk. It's a real pleasure and honor to be here. I want to thank Rachael, NBTS, David Aarons, and Kirk for having me. So I was asked to talk very briefly about precision medicine, which is also known as targeted molecular therapy. And this treatment has really transformed therapy for some types of cancer over the past 20 years. And it has been made possible by our knowledge of the structure and workings of DNA. So as most of you know, Watson and Crick discovered the structure of DNA in the 1950s, and they won the Nobel Prize for that. They described the DNA as a double helix, and the DNA is a molecule that contains the genetic code of all organisms. The DNA is transcribed to give rise to a molecule called RNA, and this in turn is translated into proteins that are critical for the working of the cells in our bodies. So I'm just going to touch on genes and the progress that has been made. So what is a gene? Genes are part of the DNA in our cells. In each of us, we have 20 to 30,000 genes in our DNA. Every cellular process, including the growth of cells, is based on the genetic information and the production of a proper protein. If these genes become damaged or mutated and they direct the production of the wrong protein, then you can get disease. And if these genes control the growth of the cell, then the cell can grow uncontrollably and you can end up having a cancer cell. So why is this important? It's important because if we can identify the damaged gene, understand what mutations there are in these genes, we can develop drugs to block the effect of that damaged gene and prevent tumor from growing or restore health in other ways. Over the past 2 decades, there has been tremendous progress in our ability to determine the molecular changes in these genes and to understand the mutations. This has also been helped by the fact that the cost of sequencing has decreased dramatically over the past 10 to 20 years. It costs $1 billion to sequence the first human genome. But now many brain tumor centers, including ours, can routinely screen a patient's tumor and look at the 500 most important molecular alterations for a cost of less than $1,000 per sample. So why what can we do with this information? We know that with most tumors, including glioblastomas and other types of brain tumors, that the tumor in each patient is not the same. They can have different molecular alterations. And if we can study the molecular alterations in each of these tumors, then we can identify a group of patients that are predicted to have a good response to drugs or combination of drugs. We can identify patients that will not be helped with that particular drug so that we don't expose them to a treatment that's not going to be helpful and may have side effects. And we can potentially identify patients that are at increased risk of having side effects and again, not expose them to the drug. Looking at this in another way, if you have a patient with a brain tumor that has mutation A, this is a different patient with a different mutation B, and this third patient has a different mutation C. So you don't want to treat them all with the same drug. Patient A should receive a drug that's specific for that mutation, same with patient B, and same with patient C. So in this way, we can personalize the treatment for each patient and give them the drug that's most likely to be helpful and the least likely to cause side effects. For some cancers, this has really made a dramatic difference in outcome. One of the first targeted molecular drugs to be introduced was a drug called Gleevec, which was introduced over 20 years ago for a rare leukemia called chronic myelogenous leukemia. At that time, this was a serious condition that shortened the lives of the patients that were affected by it, and that you can see over time with the introduction of Gleevec and then with subsequent generations of drugs that worked in a similar way to Gleevec. The vast majority of these patients with chronic myelogenous leukemia now live a fairly normal lifespan. And so we've converted a cancer into a chronic condition that can be managed in the same way that high blood pressure or high cholesterol can be managed. And the hope is that we can find similar treatments for brain tumors going forwards. There has also been improvements in other cancers. There have been drugs for HER2-positive breast cancer. With a skin cancer melanoma, there's this BRAF V600E mutation, and there are drugs that cause shrinkage in the vast majority of these patients if they're treated and improve survival. And the same is true for lung cancer. And these same drugs are now working for brain metastases. In the past year, there have been exciting developments with drugs that cross the blood-brain barrier and can treat HER2-positive breast cancer brain metastases. The same drugs that are useful for BRAF-mutated melanoma can also work for brain metastases. And in lung cancer, there are specific mutations that also respond to these drugs. So I think in the past few years, there has been real progress in the use of these targeted therapies. The situation in brain tumors is a little bit more complicated. This is a diagram that shows receptors on the surface of the tumor cell that send signals through various pathways to tell the cell to grow. And one of the receptors on the surface of glioblastomas, for instance, is the epidermal growth factor receptor, which can be present in multiple copies, sending more signals and telling the cell to grow. And you can block these receptors or these pathways, but the tumor will sometimes learn to signal through a different pathway. And so potentially you have to hit multiple pathways to turn the cell off. Otherwise, you could be playing a game of whack-a-mole. The other challenge in brain tumors is that there's a condition called heterogeneity, where the molecular alterations in a tumor in one part of the brain may be different from the molecular alterations in a different part of the brain. And so potentially you would need to use drugs to target both alterations, and so you would need to use combination therapies. Despite all these challenges, there have been some drugs that have shown activity in brain tumors. Perhaps the best known is a drug called bevacizumab or Avastin. This targets something called VEGF, which is made by the tumor. Tumors need blood vessels to bring in food and oxygen to help it to grow, and the tumor makes VEGF, which stimulates the growth of these blood vessels. It also is the main cause of the swelling around brain tumors that causes such a lot of symptoms. And bevacizumab binds VEGF, so it reduces the swelling, and it makes it harder for the tumor to grow and make new blood vessels. And this is a patient I saw in clinic just last week, showing the change you can see with bevacizumab over the course of 6 weeks in terms of tumor size and in terms of decrease in the swelling. Another targeted therapy involves targeting the BRAF 600E mutation that is common in melanoma, but is also present in a minority of patients with glioblastomas and other lower grade gliomas. Using a combination of drugs that target this pathway, dabrafenib and trametinib, about a third of glioblastoma patients have shrinkage in the tumor, and the shrinkage can be quite durable, lasting for several years. Another important target that has been identified recently is in these rare high-grade gliomas that involve the middle of the brain. They're called midline gliomas, and they often have a mutation called H3K27M. And there's a drug called ONC201 that has shown responses in some of these very difficult to treat tumors. And the development of this drug is something that NBTS is helping with. There are other rare molecular changes that respond to therapy. Recently, it has been described that NTRK fusions, which are again present in a small minority of both adult and pediatric brain tumors, respond dramatically to NTRK inhibitors, and there are a couple of these available. And then finally, although I've shown you examples of tumors that respond to one drug that hits one target, I mentioned before there's this problem of redundancy of signaling pathways. And so if you had a drug that hit multiple targets, that might potentially also be a good thing. And so one drug that's being investigated is a drug called regorafenib that hits multiple targets listed here. It has shown activity in a phase 2 trial in comparison to lomustine, and it's being developed more extensively, potentially for FDA approval in a large international trial called GBM Agile that NBTS is also helping with. And then finally, there's been progress not only for high-grade tumors, but also for low-grade tumors. Most low-grade gliomas, grade 2 and 3 gliomas, have an IDH mutation, and there are many IDH inhibitors now being tested in these tumors, and some of them are showing promise, and one of them, a drug called vorasidenib, is going for FDA approval in a large international trial. So I think in the past few years, there has been significant advances in understanding the molecular alterations in brain tumors, and this has allowed precision medicine to be feasible for some brain tumors, and some of these drugs are showing promise. There remain challenges of getting drugs through the blood-brain barrier and dealing with the redundancy of signaling pathways and tumor heterogeneity, and a lot of the focus of the current research is to address some of these issues. But there's been real progress in recent years. So thank you.
Well, thank you, Dr. Nguyen, for that really fantastic overview. Really great. Before we dive into some questions, let me just further highlight some of the additional items about the National Brain Tumor Society's efforts in our Defeat Brain Tumors platform that really kind of focus in on these precision medicine approaches. So first, as Dr. Nguyen showed, we have this molecule, ONC201, and this is an experimental treatment from a company called Oncoceutics and Chimerix. And as Dr. Nguyen showed, it's showing some potential benefit for both pediatric and adult high-grade glioma patients that have a specific mutation. This is this histone H3K27M. So while that's very promising, it's a very low percentage of patients have this mutation. So NBTS is now funding a phase 2 trial to try to find additional patients that might benefit from ONC201. And this is focusing on a specific molecular target, which Dr. Nguyen actually referred to on one of the previous slides. In this case, it's low or no expression of a receptor called epidermal growth factor receptor, or EGFR. So another example of NBTS is funding of a personalized brain tumor vaccine trial that's based on each patient's tumor signature and combining a treatment there with a type of immune cell called a dendritic cell, along with a checkpoint inhibitor like Keytruda, which has shown so much success in previously other hard-to-treat cancers. So, the hope is that this individualized and personalized type approach, a precision medicine approach, can activate and stimulate the patient's own immune system, for hopefully a really meaningful and sustained benefit. And then finally, as a final example, and Dr. Nguyen also alluded to this, I'll mention about NBTS's collaboration with GCAR, which is the Global Coalition for Adaptive Research. That's the organization that's running the GBM Agile trial. And Agile stands for Adaptive Global Innovative Learning Environment. And this platform, it's a global adaptive trial for GBM where multiple experimental treatments can be run simultaneously. And there's a really strong focus on understanding the biomarkers and the molecular genetics for subtypes of patients. And so, for example, Dr. Nguyen showed regorafenib that is a little bit broader because it can target multiple targets that may be impacting the GBM. But there will be other ones that will be more focused. And so, again, for all of these above mentioned projects, I'd be happy to connect with anybody who's interested to learn and provide more detail. But for the sake of time, I'd like to hand it back to Rachael to be able to go over some questions. Rachael?
Thank you, Kirk. And Dr. Nguyen, thank you so much for your presentation. So before the recording, we reached out to our community to gather questions that they would most want to ask regarding precision medicine and how it could directly impact their experience. So the first question that we have for both of you is, what would you recommend to a newly diagnosed patient and their caregiver who is watching this and wants to be sure precision medicine is a part of the consideration for their treatment plan?
I think when a patient is diagnosed with a brain tumor, the first treatment is surgery. And it's critical at that surgery that enough tissue is taken, not just for the pathologist to make a diagnosis by looking at it, but also to have enough tissue for two things. One is to potentially send to a lab to do the sequencing so we understand the major molecular alterations that's driving that tumor to grow, and potentially allow the patient to join clinical trials either at diagnosis or at a future date. And then also to have enough tissue that the patient can actually join clinical trials, because most clinical trials will require a certain number of slides. This is important, not just when the patient has a craniotomy and a large chunk of tumor can be taken out, but it's especially important if the surgeon is thinking of only doing a biopsy, because if the surgeon keeps the need for tissue in mind, they can actually safely do a biopsy but get enough tissue so it doesn't limit the patient's options going forwards.
Thank you very much. So we have another question, and I think part of your presentation may have answered this, but because it's a question from our community, can precision medicine be used with all tumor types?
So I think each type of brain tumor is driven by different molecular alterations. And even the same tumor, like a glioblastoma, will have different molecular alterations driving it in different patients. And that's why it's important to sequence the tumor to understand which are the major molecular changes that are driving tumor growth in that patient, and then choose specific treatments for that patient. And it may be different from one patient to another. That's why it's so important to be able to do the sequencing.
Dr. Wynn, I thought of something as you were talking about this. What about those instances where you have a midline glioma where surgery may not be an option? Is there any way to, go about trying to figure out the characterization there?
I think nowadays surgeons are really good. And so at a good center, even the midline gliomas can usually be biopsied. But the other area where there has been some advance is to take spinal fluid, and sometimes you can sequence small amount of tumor cells or DNA in the spinal fluid to understand what molecular alterations there are. Eventually, we would like to be able to take a patient's blood and figure out what the tumor is made up of. That's possible for other cancers in the body. But right now for brain tumors, the amount of DNA in the blood is so small, it's a challenge still to get a good read.
No, it's interesting. And maybe potentially with the advent of focused ultrasound being able to transiently open the blood-brain barrier to get more biomarkers out, maybe there's a possibility there.
Yeah.
Thank you. So the next question that we have gathered from our community is, how do I know if my current hospital offers genomic testing? And the second part of their question is, what if they don't?
I think most of the time your physicians will tell you if they're going to do this because they would ask your permission to do this. But you should just ask. And if they have it, and there's enough tissue, it'd be a good thing to do. And if they don't do it routinely, then you can talk to your clinicians and see if they would be willing to send it off. There's a lot of commercial firms that also do this sequencing.
Excellent.
Just to add one more thing.
Absolutely.
With some gliomas, having the IDH mutation has always been important, but it's actually going to be even more important because there's a new grading scheme for gliomas coming out in the next month or two. And so knowing whether your tumor has an IDH mutation becomes really crucial. And sometimes you can determine that just by staining the tumor for IDH. But in some tumors, it's negative by staining, and then you have to sequence. So the need for sequencing has become even more important.
Maybe I'll just add on there because, if you're a lot of the top neuro-oncology centers like Dana-Farber and UCSF and Sloan Kettering, they'll have their own panels, MGH, and you can get it. But as Dr. Wynn mentioned, there are other options there for, Foundation Medicine, Caris, Illumina, and others. So just there are other options for getting this done.
Excellent. Thank you. So the next question is one that, does not surprise me at all is being asked, and that is, how is this paid for?
Yeah, that's an important question because understanding the IDH mutation is such an important thing. There are a number of states where insurance companies will cover it. And the Society for Neuro-Oncology and the people in the NCCN guidelines are trying as hard as they can to make the case that sequencing for brain tumors is really part of standard of care. And hopefully, eventually, everybody will have this covered as routine care.
Yes, I would agree with that, for sure. The next question from our community is, should I have a second opinion on those testing results?
I think most companies and most brain tumor centers, if they have their panel, it's pretty accurate. One of the things that limits how good the testing is really the purity of the specimen. So if the specimen has a lot of tumor, then you get a better read. If it's mixed up with a lot of dead cells, then the read is not as good. But usually you don't have to repeat the tests because the tissue is so precious. You don't want to be just duplicating tests if it's not going to make a difference.
It's really interesting. I was interpreting the question a little bit differently, and it was more along the. So I completely agree with you about the reliability of the test. I was thinking more about the analysis of the molecular genetics, and for me, I would say for sure it'd be great to get more than one opinion on, the implications of those, whatever the profile looks like.
There are some reports that will suggest some clinical trials if you have a particular molecular alteration. Those reports are often written by folks who are very good but not actually focused on neuro-oncology. So frequently those trial recommendations are actually not accurate. So in that sense, Kirk is absolutely right. Trying to decide what to do with those results, I think it's useful to get a second opinion.
For our community who would be facing recurrence, do the testing results change if their tumor has recurred?
Yeah, that's a great question. And it until last year was a major question in the field that was unanswered. But there have now been several very large studies where centers from all over the world have collaborated to understand how the molecular alterations at diagnosis changes compared to recurrence. And I think the good thing is that there are some molecular alterations that probably don't change much at all, like EGFR amplification, which is a major change in glioblastoma. There are some alterations like EGFR mutation that do change. So the implications vary depending on what your treatment is. If you're targeting something that's unlikely to change, then you can probably use the tumor from the initial diagnosis to guide you. But if you're targeting something like EGFR mutations that often may disappear at recurrence, then theoretically, to get a better idea, you would have to do another biopsy or surgery and examine the tissue to know for sure that it's in the recurrent tumor. The challenge is that these tests takes time. It's anywhere from 2 weeks to 4 weeks. And for a recurrent tumor, there's time pressure because you want to get on with the treatment. So sometimes it's not feasible to wait for the results of the retesting.
Another question from our community deals with, and this is an interesting question. If I did not have this testing done at the time of my diagnosis, how can I get it done now?
Yeah, it can be done anytime. So if you've had the diagnosis a while ago and the tumor is stable, but you have tissue available, it could still be done. And in some ways, it's not a bad thing to do, because if you wait until there's a recurrence and you want to get on with treatment, you may or may not have the time to wait a month or so to get the results back.
Okay. That is most of the questions that we have from our community. And, part of Informed in 30 is that this recording is provided to our community in 30 minutes. What I wanted to ask as we start to wind down is if there is anything else that either one of you wanted to add before we begin to close down.
I think there's been a lot of challenges in developing precision medicine for brain tumors, but I think in the past couple of years especially, there has been really encouraging advances. And NBTS has really helped tremendously in moving that progress along. So we're grateful.
And I guess the only thing I would add is just that we're also very grateful for our partnership and our collaboration with you, Dr. Wen, and Dana-Farber, and the whole community, and really coming together and figuring out how to use these new precision treatments in whatever shape or form they end up being. I think we are getting close. We're getting closer, but there's a lot of urgency. And so we really have to figure out how to do this in the most efficient way. So again, we're pleased to be able to bring these informed in 30 things to you. And I think, with that, I'll hand it back to Rachael, and she can tell you about potentially what our next one's going to be. And thanks again, Dr. Wen. We really appreciate it.
Thanks so much.
Yeah, truly. Dr. Wen, thank you for joining us today, for creating such meaningful content for our community, and for answering the questions that also will help them to better understand how precision medicine could impact their experience. And of course, Kirk, thank you so much for all that you did to prepare for this for us today. I would like to thank our community for joining us to be informed about innovations in precision medicine and what that could mean for you. Please join us in June for our next installment of Informed in 30, Innovations in Surgery Techniques with Dr. Eja Endom from Emory University. Thank you very much.